Thank you so much for showing up at 8:30, and everyone was there on time, and it was great. And I actually really like there's a bit of delay because now I've had a chance to wake up, and otherwise it wouldn't have gone very, very well. So thank you so much for being here, and thank you to Deborah and Graham and everyone in the ALK community for making this possible and putting me on this stage, literally. Yeah, and I'd just like to start with a big thank you and to the ALK community, and I wouldn't be here without your support and encouragement. So today I'm just going to quickly go through my PhD project, which I'm starting on Monday, and just let me know if at any point there's something that you don't get, because your time is valuable, and I really appreciate you being here. And this is a great chance for you to give me feedback, and then I can do what you think is most important for my PhD. So I've got the money from CRUK, Cambridge Center, and I can kind of decide what I want to do for my project. So if there's anything that I haven't mentioned that's really, really important to you, this is a great opportunity for you to really decide what you want to see in research. So yeah, just let me know if at any point I've lost you. Great, so I'll be talking about my project, which is understanding ALK and overcoming resistance to ALK TKIs in ALK positive non-small cell lung cancer. So I'll just start with a little bit about me. So my dad was diagnosed with lung cancer, ALK positive lung cancer in 2022, when I was a second-year medical student at Imperial College. And because of this, and he had an aggressive disease, and he kept on progressing, and he wasn't a typical ALK patient, I got really curious about why he's not responding like everyone else. So I decided to take some gap years out of medical school, and I first did an intercalated bachelor's in Cancer Frontiers at Imperial College on how to predict response to ALK TKIs, because that's what was most relevant to me. And then now I've decided to take 4 more years out of medical school for a master's plus PhD in cancer biology at the University of Cambridge. So during my master's, I've looked at 2 main things, so understanding the effects of ALK TKIs and how to overcome resistance. So this is what I'll be continuing throughout my PhD, what I think I'll be doing, but maybe I'll change my mind. And I'm at the very start of this, so I'm starting on Monday. So hopefully, what I'm hoping to get from this PhD is a better understanding of ALK TKI side effects, and possibly how we can manage them better, and identifying some bypass resistance mechanisms, how to overcome them, and some combination treatments, and maybe that will lead to a clinical trial. So that's the best case scenario from a 3-year PhD. I'm really shooting for the stars there, so if I come back in 3 years and I haven't achieved this, don't expect too much. So I'll just go through some biology, just to make sure everyone's on the same page. So here's a picture of the cell. We have the cell membrane and the nucleus. And inside the nucleus, we have the DNA, which is the coding, the instruction book for everything that the cell does. And it gives out these instructions by making RNA, which we call messenger RNA. And this protein, this piece of protein, is able to move from the nucleus into this bit of the cell called the cytoplasm, where we all have the industry, the production machines of the cell. And through this RNA, the cell is able to tell, okay, the DNA wants me to do this, and produces the protein accordingly. Okay. So the ALK receptor is a type of protein. And the ALK receptor is present on a lot of cells, normally in the normal adult healthy person. And usually it's not active, so it's just sitting there and not doing anything. It only gets activated when another protein binds to it. And the protein that binds to the ALK receptor in normal context is called alkal-2. So it's a smaller type of protein. And it activates that receptor. And we know a receptor is activated when a phosphate group attaches to it. So I've represented that with the yellow P. And we call this process phosphorylation. So now we know, okay, the receptor is active, and it's able to tell the cell what to do. So for the ALK receptor, it will tell the cell that it's time to grow. So in lung cancer, the most common fusion that we know is EML4-ALK. So usually these 2 genes sit separately on the chromosome 2. So they're not touching each other. But when the cell is replicating, sometimes mistakes happen. And the ALK EML4 gene gets switched around, and they get stuck together when really they should be separate. And because now the DNA is a bit messed up, the protein that's going to be made from the DNA is also going to be abnormal. So we get this fusion protein between the ALK and the EML4. And because of this fusion, the ALK protein is always activated, so it always has this phosphate group. And because of this, it's always telling the cell, it's time to grow, it's time to grow. And you got this uncontrolled cell growth that leads to cancer. So we know from Professor Povat's talk and through this weekend that since the discovery of EML4 ALK in lung cancer in 2007, there have been amazing classes of drugs. So 3 generations of ALK TKIs, a lot of people here on that. And there's a 4th generation coming along called neladalkib in the phase 3 trial. And all of them work in similar ways. So they bind to where the phosphate usually binds, and it displaces it from the ALK protein. And because it's no longer active, it stops the cell from having that signaling to grow. And a cancer cell always needs that message in order to be able to survive and grow. And if you cut that off, it dies. So we've seen how amazing each generation is. You know, they're more and more potent, more and more brain penetrant. So this is from the CROWN trial. I think we've seen this graph yesterday, with patients on lorlatinib in blue performing a lot better, so without disease progression compared to patients on crizotinib. So I'm sure everyone already knows how to read this graph, but just quickly, this is the duration patients are living without disease progression. And here we have the percentage of patients. So at time point 0, everyone's alive without disease progression. And this starts dropping off the chart as time goes on. So at 12 months, about 20–30% of patients on crizotinib are still alive without disease progression. But for lorlatinib, this sits around 70–80%. So in my PhD, I'll be using these 4 drugs because they're the most commonly used ones. And I'm anticipating that neladalkib is going to come on the market. So I want to study that drug as well and stay a step ahead. So we've seen how great they are, but there's still some challenges. One of them being the myriad of side effects that everyone in this room can tell me about. And this can reduce your quality of life and can lead to treatment discontinuation, which we don't want. The other problem with this is ALK TKIs are not able to overcome certain types of resistance, so what we call bypass resistance mechanisms, which I'll get to in a bit. So I'll start with all the side effects. I've highlighted the most common side effect per ALK TKI, and you can see they're all a bit different. They're not exactly the same, and everyone here will have that experience. Especially with lorlatinib in blue, you can see the blue squares are a bit different from the rest of the TKIs. And you know, the brain side effects, you have weight gain, peripheral neuropathy, all that kind of stuff. But really, we don't know why ALK TKIs are causing these side effects. We think it's because they're inhibiting ALK, but we don't really know why it's causing heavy legs, or why it's causing photosensitivity, so rashes when exposed to sun. We don't know why. And because of this, we don't know how to manage them properly. And we also don't know how to design less toxic drugs. And all of this is because we don't understand what ALK does normally in the body. So on the left, I've shown a little picture. And this is where ALK is normally expressed in the healthy adult human who doesn't have cancer. And you can see actually ALK is expressed in a lot of tissues. So it's in your muscles, in your fat, in your bone marrow, your lymph nodes, your lungs, your brain. So this is normal. ALK is supposed to be expressed in these tissues. And because of this, it's only recently that we found that actually ALK is associated with a myriad of other diseases, not cancer. So it's associated with sepsis, with obesity, with pain, Alzheimer's disease. And this is all very new research that people are working on, because up until now, we still don't know what ALK does. And this is what I'll be looking at in my aim 1 of the PhD. What does normal ALK do? The other bit that I want to look at is resistance to ALK TKIs. So it can be broadly divided between off-target resistance and on-target resistance. So I'll start with on-target resistance. So on-target means that the problem with this resistance is because of ALK, the target. So usually we have a point mutation, so a single mutation within the ALK protein, and it prevents the ALK TKI from binding. So this leads again to cell growth and an active ALK. When it comes to off-target, and in my case, I really want to study bypass resistance mechanisms, the ALK inhibitor is still working fine, but on the cell surface, you got so many other receptors. So a common one might be EGFR. And this can get activated. And this will lead to cell growth and the signaling again, bypassing ALK inhibition. And these bypass resistance mechanisms is really, I think, a growing priority in our community. So this is the profile of patients who have progressed after second-generation TKI, so alectinib, brigatinib. And 40 to 50% of patients are WT, so that means wild type, so ALK wild type. And that means that ALK is the same one that we find in nature, in the wild, so it's normal ALK. So these patients, they're progressing, but they have normal ALK. So the resistance mechanism is by definition off-target. And the most common type of off-target resistance mechanism is bypass resistance. After a third generation, so lorlatinib, we see that this proportion of patients, so 40-50% wild type, has increased to 50% wild type. So more and more patients in the future, I expect, will not be having ALK mutations, but will be having these off-target mechanisms. Because the ALK TKIs are working so well, they're doing what they're supposed to do. So in order for the cell to survive, it has to find different ways to do that, and it's not through ALK mutations. So this is confirmed by a real-world study in June 2024, and they've looked at what patients have when they progress. So most patients at that point still are having secondary mutations, so on-target mutations. And others are squamous cell transformation, KRAS, BRAF, MET, so we've all heard of this. But the pink people, that's 25%. 1 in 4 patients have undefined resistance mechanisms. We don't know why they're progressing. And I think this is because we're using next-generation sequencing at disease progression, so liquid biopsies and things like that. And they can detect mutations, but they can't detect if the receptor is overactive. So you can tell you, okay, we know this amplification, there's a mutation in KRAS. But if it's still the same receptor, it's just more active, currently we can't detect that. And so this is something that I want to focus on and change for clinical practice. So before I dive into what exactly I'll be doing in the PhD, I'll just quickly go through the techniques I'll be using. So again, we have the cell with the DNA producing RNA, and that tells the cell what to make in terms of protein. I'll be doing RNA sequencing to answer the question, what genes are active? Because only active genes will tell the cell what to do and produce the RNA. I'll then be looking at what proteins are active by specifically looking at that phosphate group on the protein. And so this is called phosphoproteomics. And this will tell me what proteins are active. And by combining these two techniques, I get a good picture of the cell and everything that it's doing, both in terms at the genome level and at the protein level. So I know what the DNA is trying to tell the cell to do and what the cell is actually doing. So in my first aim, so understanding normal ALK, I'll first start by stimulating the normal ALK receptor in the adult mouse, because that's the closest we get. And I'll give them an injection of ALK-2, which is the activating protein for the ALK receptor. And hopefully, we'll see by RNA sequencing and phosphoproteomics what genes become active and what proteins become active. And I hypothesize that what happens when ALK is stimulated in the liver is going to be different from what happens when I'm stimulating ALK in the lungs or in the brain. And this might inform how ALK tumors form and what are the mechanisms that prevent ALK tumors from forming because I'm stimulating ALK. So I would expect that in the normal mouse that's not developing cancer, it has some ways of suppressing that. And then I'll be treating them for 6 months with either alectinib or brigatinib, both second-generation TKIs, to compare them head-to-head. And I'm treating them for 6 months. So usually, so we've seen yesterday from that talk on clinical trials, that in preclinical studies, we're testing drugs on mice, but we're only seeing, does it actually decrease the size of the tumor? We don't really look at what are the long-term side effects. And we know that patients are taking these TKIs for 10 years, 15 years. So it's really important to understand what happens on the long term, not just what happens in the first 3 months. And 6 months in a mouse life is quite a long period of time. And so that's the industry standard for long-term treatment in mice. I'll also be treating them with giving them either a control diet or a high-fat diet, because we know from brigatinib, alectinib, you're supposed to be eating this with a high-fat food. But is this actually applicable to the long term? Is it going to negatively impact your health because you're always eating high-fat foods to try and increase that concentration of drug? So this is what I'm going to be looking at. I'll also be looking by histology if there's any sign of inflammation, scarring, fibrosis in different tissues. And again, doing RNA sequencing and phosphoproteomics to see what genes, what proteins are active. And hopefully this will give me a really good picture of what happens. So let's say we see fibrosis in the lungs because of brigatinib. And I'm saying, okay, these proteins are active in the lung that has fibrosis. And this is because of these genes that are increased, or these genes that are decreased. And so I get a really good, this is what's happening, that's the chain of reactions. And because of this, maybe I'll be able to say, if you have fibrosis, we can just inhibit that protein and stop you from getting those side effects that make you breathless and make you switch treatment. Everyone's still with me? Okay. So in my second aim, I'll be looking at bypass resistance mechanisms. And for this, I'll be using a really cool technique called CRISPR activation screen. So I'm sure many of you will have heard of CRISPR already. It's won the Nobel Prize in 2020. And it's really giving all scientists a God complex, because we can do whatever we want. So we can give a mutation, remove a gene completely from the genome, and at certain times of the day, and just decide what we want to do with that DNA. So what I'll be doing is I'm going to overactivate each gene, coding gene, in the human genome. So each cell, lung cancer cell, both variant 1 and variant 3, will get one overactive gene. So I get this huge pool of cells, and I'll be treating them with either alectinib, lorlatinib, or neladalkib for 2 weeks. And at the end of those 2 weeks, the cells that have survived are the ones that are resistant, and I'll be able to look at what genes are overactive in those cells to identify the genes that give you resistance. But you might imagine, so we have 23,000 genes that are active in the human genome. From this, I might get 5,000, 1,000 genes that can give you resistance, but I'm not going to be spending the next 3 years then testing each one of those genes to see which one is the most important. So I need to know which ones are actually relevant to patients, and this is where you come in. So there's a trial that's been set up at the Royal Marsden called the Festival Trial. The lead investigator is Professor Popat, so I'll be working with him. And patient samples will be sent to me when you get a biopsy, both either from the blood or tissue, will be sent to my lab, so the Turner lab in the University of Cambridge, and we'll be doing RNA sequencing on that sample to see what genes are overactive when your disease is progressing. So this is only available at the Royal Marsden, and yeah, this is just purely for research purposes. So if we do that RNA sequencing, even if we identify an overactive gene in those patients who have generously donated their tissues, we might not be able to help them. But these patients are going to be helping future generations of ALK patients. So my third aim will be validating in vitro, so in the Petri dishes, and in vivo, in mice, the results I found in my aim 2. So what are the bypass resistance mechanisms? So if I've identified this gene, okay, I see a lot of patients who get that at disease progression, and I've also found this from my screen that's driving resistance. I'll then be verifying for sure that is the gene that's giving you resistance. So I'll be using the CRISPR technique again and giving those cells the resistant gene. And hopefully what we see is that at the same concentration here, almost all green cells, so the sensitive cells, have died. But if I've given them the resistant gene, we'll see that 50% of them are still alive. So this is how I'll be verifying, okay, these cells are now resistant to ALK TKIs. And then I'll be testing different combinations of ALK TKIs with available FDA-approved inhibitors, because I want to move this into the clinic as fast as possible. So I'll only be looking at drugs that we already have in the clinic, giving this in combination and seeing if it works in plastic plates in these cells. And if they do work, then I'll move on to mice who have ALK positive lung tumor. And hopefully what we see is that mice who have the vehicle or the placebo don't survive for as long. But with the ALK TKI, we see that they survive for a bit longer, for a lot longer. But with the combination, hopefully maybe they're cured, maybe none of them die. And that would be amazing. And hopefully from this, it can move into a clinical trial. So again, from this PhD, I'm hoping to get a better understanding of ALK TKI side effects, how they happen, and targeted management options. And then I'll identify bypass resistance mechanisms, combination treatments that may eventually lead to a clinical trial. Everyone, yeah. So I'll just give you an overview of what I've found so far during my bachelor's with Dr. Evans at Imperial College on predicting response to ALK TKIs. Then at the Department of Pharmacology with the effect of ALK TKIs on normal tissues with Dr. Merkel, who's my lead supervisor. And Professor Turner, who's also at University of Cambridge on resistance mechanisms to ALK TKIs in both ALK lung cancer and ALK positive ALCL. So that's anaplastic large cell lymphoma. It's a type of blood cancer and where ALK was first discovered in 1994, if you remember that small detail from Professor Popat's speech. So what got me interested was, well, my dad was one of those bad ALKs. But even if you look at this very famous chart, you know, lorlatinib, patients on lorlatinib, you know, don't progress as fast. But I'm really interested in that top left corner here. There's no difference between the red line and the blue line. So these patients, about 10%, it doesn't matter what drug they take, they will still progress within 3 to 6 months. So I want to know, can we identify those patients before they start treatment? Because maybe what they need is not an ALK TKI. Maybe they'll respond better to chemotherapy, or they respond to immunotherapy better, instead of just giving everyone ALK TKIs and having those 10% of patients who progress very quickly. So a value that I found in literature, in cancer literature, it's very new, is called the pan-immune inflammation value, or PIV. And it's a simple ratio. So on the top line, we multiply the number of monocytes, platelets, and neutrophils divided by the total number of lymphocytes. And the theory behind this is that the top line, so monocytes, platelets, and neutrophils, are pro-tumor. So they help drive an inflammatory tumor environment that may help the tumor invade. They might help the tumor become resistant. Whereas lymphocytes, or natural killer cells, are killing that tumor, so the anti-tumor. So when we have more of these pro-tumor cells and fewer of these anti-tumor cells, we get a high PIV number. So you would think that patients who have a high PIV number will maybe not respond as well. And so I've looked at this in 3 London centers during my bachelor's. So from 3 centers, we only get 22 patients. And I have to say, you know, some caveats first. Very small number of patients, 22. This is only the 2nd study of PIV in ALK positive lung cancer. So we can't really trust that number. We can't really trust that PIV is doing what it's supposed to be doing. The other study was in Chinese patients with ALK positive lung cancer, and they found similar things. But what we have as a problem is the cutoff score. So what does it mean to have a high PIV or a low PIV? And you can see that for overall survival and progression-free survival, that cutoff score is, you know, completely different. So if we look at progression-free survival first, patients on the right side, patients who have a high PIV in blue are different from patients who have a low PIV. They seem to be progressing faster. And we know that this is a meaningful difference because I've done some statistical tests. And in science, we call this the p-value. So usually, the p-value means we use it at 5%. So what are the chances that this difference is real? No. What are the chances that this discovery is not real if we find this difference? So usually, we limit that at 5%. So 0.05. And here, it's 0.01. So there's a 1.17% chance that this difference that we find is not real. So we can kind of trust this based on 22 patients, so not really. Whereas with overall survival, first, the cutoff score is quite different, and the p-value is, there's a 40% chance that this difference is not real. But it still looks like maybe it works still a little bit. So just take this with a grain of salt. I'm hoping to expand the study by working with Dr. Fabio Gomez and the NOTCH, which is the National Oncology Trainee Collaborative for Healthcare Research, to hopefully establish a nationwide ALK positive patient database for us to access and look at the data and see if we're able to maybe get 200 patients, 300 patients, and validate this score. Either prove that it's not right and we can't use it, or prove that it is right, and maybe we can integrate that into clinical practice. So my other project with Dr. Merkel at the Institute of Metabolic Science was on normal ALK. So ALK was found to be associated with obesity, which is really strange. So normal people who sometimes just don't have the ALK gene are resistant to weight gain. And they wanted to check this in mice. So they found that mouse who lack the ALK gene, ALK minus minus on the right, are much thinner than their sibling who have the normal ALK gene. And in the blue line, you can see that they're resistant to weight gain on a high-fat diet. They have conserved lean mass, muscle mass, but reduced fat mass. So this was found in 2020. And another group thought, maybe it's not the ALK receptor. Maybe it's the protein that activates the ALK receptor. And so they've created mice that don't have that protein that activates the ALK receptor, augmenter alpha, ALK2. And again, the mouse on the right who does not have this ALK activating protein is thinner than their siblings who do express it. And again, with reduced fat mass, conserved lean mass, and reduced body weight in general. So my group has decided, we already have an ALK inhibitor, why don't we use it? And they tried treating obese mice with an ALK inhibitor, ceritinib. And so in the gray line, mice, are gaining weight. With semaglutide, which is our Ozempic, they're losing weight, the weight loss drug. With ceritinib, in the yellow line, they're losing a lot more weight than with just semaglutide. And if you combine the two of them together, these mice are losing a lot of weight. So this is very different from the patient experience, right? What we hear a lot is actually I'm gaining weight on these ALK TKIs. But what we see in obese mice, they're losing weight with ceritinib. And there's a real question as to where this happens. So does it work the same way as semaglutide, so inhibit the brain? They're eating less, so appetite suppression? Or is it working directly in the fat? So this was my project in my master's. And a research associate in the lab, Cortina, has found that the fat is dissolving directly with ceritinib. So this is a glycerol release assay. And glycerol gets released when fat cells die and dissolve. And you can see with ceritinib and brigatinib, there's more of that glycerol release, so more fat cells are dying than with ALK2, the activating protein. And what might be more interesting is that ceritinib concentration is highest in fat, in adipose tissue, much, much higher than in the blood or in the brain. So what does this mean for patients? Does it mean that if you have a higher percentage body fat, maybe you don't need to take as high a dose because you have more reserve? Does this mean that maybe you can skip a dose and you'll be fine because this fat is releasing? Or is it just storing it? We don't know. So this is what I'll be continuing as well in my PhD. In the Department of Pathology with Professor Turner, I've looked at resistance mechanisms. So they're specifically interested in bypass as well. And they found in childhood cancers, neuroblastoma, which can also be ALK positive, and ALCL, lymphoma, the blood cancer, that FGFR, fibroblast growth factor receptor, can be overexpressed. And the good news is that we have an inhibitor that can inhibit this receptor called erdafitinib. So I thought, okay, maybe this is something we're seeing in lung cancer as well. Let me try it out. So I decided to try this in both variant 1 and variant 3, because we know about a third of patients of variant 1, a third of patients of variant 3. And they can be represented by cell lines, so cells that are growing dishes that are readily available, and lots of labs have used them before. So for variant 1, this is called H3122, and variant 3 is H2228. And what I found is that inhibiting FGFR in combination with brigatinib seems to work better in variant 3. So these two plots are called synergy plots. So synergy is when two drugs are working together. They're not just adding on top of each other, but they're cooperating in some way. So you can use a lower dose of each drug, but kill more cells. And the more red it is, the more synergistic it is. So it is more red in H2228, which is variant 3. And I confirmed this by treating them and looking at how many cells are alive at the end of this. And you can see on the right that variant 3, a lot more cells are dying with the combination treatment than with variant 1. And this could be because variant 3 might be more reliant on FGFR signaling. So this is a paper from another group, Furugaki. And so if we look at the graph on the left first, the black lines are ALK TKIs, alectinib and lorlatinib. So with increasing concentrations of the drug, more cells die. And they're not sensitive at all to the blue line, which is our FGFR inhibitor. But when they become resistant to ALK TKIs, so the second graph, you see this flat line almost with ALK TKIs, they become very sensitive to FGFR inhibitors. And once they become resistant to this FGFR inhibitor, now we see a reversal. They're now resistant to FGFR and sensitive to alectinib again. So this was only seen in variant 3. So it seems like variant 3 has this ability to kind of do whatever is most convenient for them. You're inhibiting my ALK? Fine. I'll just use FGFR instead. And it just switches between these two states. And it's allowing them to maybe survive better. And this is maybe why we're seeing variant 3 patients progressing faster, because variant 3 is more adaptable. And they tried testing this in mice. So this was in lymphoma, the blood cancer. And you can see that the combination-treated mice, in pink, it's a flat line until the end, compared to just alectinib in red. And they found the same thing for neuroblastoma, so the neurological cancer, that the combination treatment works better than any single treatment. Another mechanism of resistance could be senescence. So I'll explain that. When we're treating these tumors, a bunch of cells with ALK TKIs, most of them die, but you get this other population that are senescent. So senescence essentially means aged cells. So they're not able to grow anymore. They're old, and they just stay there. They can't reproduce, they can't have children. But it doesn't mean that they're not active. So they have a very active social life, and they can still tell neighboring cells how to survive. And because of this, we might get a third population of cells that are actually resistant to ALK TKIs. And what I found is that variant 3 might be more senescent, so it has more of these old cells that have wisdom and can tell the younger cells, this is how you survive. So variant 3, again, much higher proportion of senescent cells compared to variant 1 after 3 days of treatment with alectinib. And what's more interesting is when I made both of these cells of a variant 1 and variant 3 resistant to ALK TKIs, I see a higher proportion of these aged cells. So this suggests to me that these senescent cells, aged cells, are contributing to the ALK TKI resistance. I tried treating them with a drug that specifically kills senescent cells. Didn't work that well, so that's why the graph is tiny. But I'll be working on this on my PhD as well. But I've bombarded you with lots of graphs and pictures and data and all of that. But really, the take-home message is that there is hope. There are so many things that we're working on, as you have seen: senescence, FGFR inhibitors, combination treatments, and what Professor Popas mentioned, antibody-drug conjugates, PROTACs, even neladalkib, the ALK vaccine, so many things that are developing in the field. So it's only going to get better from here. And I'd just like to thank every one of you for listening, for getting up so early in the morning. The ALK community, the charity, the organizers of this event, and the wonderful people in my labs who have supported me throughout this MRes. And these are my references, and I'll leave you here.
Does anyone have any questions for Roxanne? Yes, I thought there might be one or two. There's one here. Thank you, Andy. And then there's one heading over here. Thank you.
Just as a slightly older patient anyway, does the senescent cells have anything to do with age, or is it just their older cells? Because clearly your father was not a young patient kind of thing, but does age, when you sort of discover the cancer, have any relevance to resistance to the TKIs?
That's a really good question. Yeah, so senescence is a natural process of the body. So as we age, we naturally get more senescent cells. And what's really difficult to dissect from this is that these senescent cells actually, when they work properly, is they stop cancer from growing. So at the beginning, when the oncogene is first activated, so we actually see that these cells become senescent as part of a protection mechanism. And it stops the tumor from being able to grow because now they're old. But when this tumor is already established and these cells are becoming senescent because of the treatment, there seems to be an opposite effect. So senescence is a very complicated process, and it has these opposite actions, and we don't really know why it's happening and how to tell the difference between a senescent cell and a normal cell. So there's a lot of controversy in that field. Yeah.
Roxanne, that was absolutely wonderful. I'm so impressed, and I am so hopeful that you're there doing this for us. So thank you for that.
Thank you.
I'm conscious that I've got a biopsy specimen. I know it's there somewhere, either in my hospital or somewhere close by. And I'm just wondering whether or not there is any way that those of us that would like to donate some of our, the original biopsy specimens, if they're of any use, to research, what the pathways are for that. You're clearly waiting through Prof. Popat's study to collect samples, presumably from new patients or people who are progressing. But there are lots of us here who've got ownership of biological specimens that if there was a way of getting them to you, we would probably, others than me, would be happy to do so.
Thank you. Thank you for being so happy to help me in my research. It really means a lot. And I've also wanted this because I have my dad's biopsies, and I thought maybe I can sequence them as well. And the problem with it is usually when they're kept in the lab, they become FFPE. So it's fresh frozen paraffin embedded. No, formalin fixed paraffin embedded. So because they've been treated chemically to stop the tissue from degrading, a lot of the DNA, RNA information has also been degraded. So we can't really use that information to get everything we need. But otherwise, it would have been lovely, and thank you for asking.
I'm wondering whether, in fact, thinking perhaps there may be crossover between your area of research and other areas of research that are not cancer-related at all. And in particular, billions of dollars are being invested at present in what's generally known as juvenescence or anti-aging. And part of that process, I know a lot has been invested in looking at reducing the load of senescent cells. In particular, there is a drug I know that has been identified as quite material in that process, and in fact I think it might even be a repurposed drug. So that is my main question, but just a side question as well. I'm fascinated by what you're doing, and I'm just wondering whether there's any way of sort of staying in touch and following you on your path over the next 3 years.
Right. Thank you for your questions. And yes, you know your science and all the science field around. Yeah, so anti-aging medications are definitely booming at the moment. There's a huge market for it. And one of the drugs that I tried treating the senescent cell with, the senolytic, is one of those anti-aging drugs. It didn't seem to work that well for lung cancer. So there might be some barrier there or some other resistance mechanism for it. But definitely, my future plan is to just go along and test all those anti-senescent drugs and see which one sticks. So yeah, this is what I'll be doing. And in terms of keeping in touch, I'm hoping to stay in the group and keep you updated with Deborah's blessing.
Roxanne, thank you so much for this wonderful presentation. So it's very impressive. I totally am with Gile here. So we have hope with people like you. Thank you.
Thank you.
My question is, it's more like a comment about the senescent cells. So from the literature and from previous studies, it's, the picture is more complex. So this population of drug-tolerant persisters, the DTPs, as they're called, seems to be more complex than that. So some studies show that these cells are, that could be stem cells, cancer stem cells. They could be quiescent cells, which are cells which kind of are asleep. They are out of proliferation cycle. And there was a recent study which actually shows that these persistent cells are already existing from the beginning in the population. So they're not genetically different from other cancer cells, but they're different metabolically. So obviously it adds to the complex picture. So you are talking only about senescent cells. What is your comment? How are you going to deal with this complex situation when people would start arguing that why you're only considering senescent cells?
Yeah, thank you, Professor, for that, for that very astute question. Yes, so senescence is really just one bit of the picture. We know that the immune environment around the tumor is also causing maybe some of that resistance. We think that maybe the senescent cells are driving it, but it could also be all the other cells, so the platelets, the monocytes, the macrophages, that are helping drive that environment. And absolutely, with senescent cells, they're very similar to the normal cells, and it's really difficult to identify them. So at this minute, I've only used one technique to identify them. I haven't even confirmed that they are in fact senescence. I've only used this one marker, senescence-associated beta-galactosidase. And I definitely need to do some more characteristics. I'm thinking of isolating that population and doing RNA sequencing on them to really characterize what they're doing, what they're made of, but also the proteins that they're secreting and the cytokines, the messages that they send into the environment to modulate that. But with regards to this, there are also, again, all the other reasons why there might be drug-tolerant persister cells. And actually, in Professor Turner's lab, we have some members who are actively working on identifying cancer stem cells. And we think that we're onto a marker, but happy to discuss this in private because it's confidential data, not unpublished. Yeah, a very good question, and hopefully we'll be able to identify them very soon.
Roxanne, it was a fantastic, fabulous presentation, and thanks to Deborah and team for arranging it for everyone. My question is regarding, is there any relationship between body composition, which is fat to muscle ratio, water ratio, and the resistance to TKI?
That's a really good question. So there's a paradox in lung cancer called the obesity paradox. And it seems like patients who have a higher BMI, we know that obesity is associated with mortality and all kinds of diseases. But actually in lung cancer specifically, a higher BMI is associated with better survival outcomes. So it could be because of functional reserves. So with chemotherapy and all of that, you have more to go around, so able to survive for longer. Yeah, it could be that. That's one theory. But we also know there's existence of things called sarcopenic obesity, so the muscle to fat ratio. Yes. And that's an adverse, so it's a risk. It doesn't benefit your survival. So there's definitely some kind of balance there. And we've just seen that very new data where ceritinib seems to accumulate a lot in the fat. We don't know if that plays a role because that data was, we collected that just a couple of months ago. It's not published anywhere. So we really don't know. But yes, it seems like patients who have a higher BMI survive for longer.
So just to simplify it, high BMI may not necessarily mean high fat. It may mean that you have high muscle, low fat, but high BMI, versus high fat and low muscle, which is sarcopenic, as you said.
Yes, so markers for sarcopenic obesity is through a CT scan, and they have to measure the proportion of
Yes.
muscle to fat. And for that one, it did seem like having a lot of fat, visceral fat specifically, not fat around the thighs or the
Yes.
butt. Visceral fat seemed to be bad, but peripheral fat with muscle seemed to be good.
Oh, lovely. So more strength training then.
Thank you very much. I'd like to thank Roxanne on behalf of all of us for an absolutely outstanding presentation. So I've learned so much this morning. I've learned so much this morning, as I'm sure everyone who was here did. I don't think we've seen the last of this young lady. I think she'll be at future conferences. So I would like final round of applause for Roxanne. Oh, a wolf was said. It's never happened before.